Myself, Rajendra Shetty, Assistant Professor at Ganpat Parsikar College of Education, Harmal, Goa. Title of the unit, Alteration in Chromosome Number. Module name, Euploidy. Outline, Heteroploidy, Euploidy, types of Euploidy. Learning outcomes, understands heteroploidy and its types, defines euploidy, understands types of euploidy, heteroploidy. Chromosomal aberration, which results in change in number of whole chromosomes, is called heteroploidy. Heteroploidy is basically classified into two types, euploidy and aneuploidy. In euploidy, there is loss or gain of whole chromosome set. In aneuploidy, there is loss or gain of part of chromosome set, euploidy. If any species contains multiples of basic chromosome set, then such species are referred as euploids and phenomena is referred as euploidy. Example, a plant with a basic chromosome number 7 may have euploids with chromosome number 7, 14, 21, 28, 35 and 42. Types of euploidy. Euploidy is classified into three types, monoploidy, diploidy and polyploidy. Monoploids. Monoploid species have single basic set of chromosomes. Monoploidy is common in plants and rare in animals. For example, in barley, 2n is equal to 1x is equal to 7. Origin and production of monoploids. In angiosperms, they may occur naturally due to parthenogenetic development of egg. They may also be produced artificially, mainly by distant hybridization method and entoculture method. Morphology of monoploids. Monoploids are generally smaller and weaker than the diploids. They have reduced size of all vegetative and floral parts. The size of seeds, stomata, and diameter of pollens are found to be smaller than their diploids. They show low viability. Their nuclear volume is less than their diploids. Cytology of monoploids. In zygote stage, pairing of chromosome is not possible. In metaphase 1, chromosome appears as univalence. In anaphase 1, each chromosome moves independently of the other and goes to either of the two poles, as seen in the figure. If meiotic process succeeds at all, the univalent chromosomes are found scattered all over the cell. Thus, chances of formation of functional gametes are very rare. Uses of monoploids. Monoploids carry a single set of genes, so they are genetically pure. Thus, they can be used experimentally to a good advantage in crop improvement. They are used for developing homozygous diploid lines. They may be useful in isolation of mutants because the mutant allele, even if recessive, expresses itself. Polyploidy. An organism having more than two basic sets of chromosome is known as polyploid, and the phenomenon is known as polyploidy. Half of total species of flowering plants are polyploids, and 2/3 species of all grasses are polyploids. A typical diploid is rare in nature. Many of our cultivated diploid plants are actually polyploids. Origin of polyploidy. Doubling of the chromosomes during early stage of development of the embryo can result into polyploidy. Also, union or fusion of the unreduced gametes or union of two male gametes with a single egg can result into a polyploid. Due to spontaneous mutation in nature, polyploidy can occur. Also, it is induced by the plant breeders. Types of polyploids. Polyploids can be broadly classified into two types, that is, autopolyploids and allopolyploids. Autopolyploids have multiple sets of same genome. Allopolyploids have multiple sets of different genome. Autopolyploids. Autopolyploids have more than two copies of the same basic set of chromosomes. For example, if X represents basic chromosome set, then 3X will result in autotriploid, 4X will result in autotetraploid, and 5X will result in autopentaploid. Origin and production of autopolyploids. They may occur naturally or may be induced artificially. In nature, they may arise as a result of interference with cytokinesis or unreduced gametes produced during meiosis. Autopolyploidy may be induced in plants and animals by treatment with chemical mutagens like colchicine, radiations like X-rays, and temperature shocks. Morphology of autopolyploids. They are larger in size. The leaves, flowers, and fruits are large but usually less in number. The guard cells are larger and have large frequency of stomata per unit area. Pollen grains are larger and the cell contains more chloroplasts. Autotriploids. Triploids have three similar genomes and are commonly designated as 3N plants. It occurs in number of plants like Oenothera, Datura, rose, watermelon, banana, sugar beet, tomato, grapes, etc. Origin of autotriploids. They originate by hybridization between autotetraploid and diploid, as you can see in the shown figure. Characters of autotriploids. They are more vigorous than normal diploids. They are leafier and show tendency towards perenniality. Sometimes shows floral abnormalities. Their pollen grains, stomatal guard cells, and wood cells of xylem are larger than those of diploids. Plants are highly sterile and seeds are rarely formed. Cytology of autotriploids. The meiosis is highly irregular in autotriploids. During synapsis, three homologs cannot orient themselves so as to give equivalence at the two poles. Meiotic products may contain either N plus 1 or N or 2N chromosome. As you can see in the figure, the gametes arising from triploids have unbalanced genome. Autotetraploids. Autotetraploids have four similar genomes and are commonly designated as 4N plants. You see them in rye, corn, red clover, snapdragon, allium tuberosum, etc. Origin of autotetraploids. Autotetraploids may originate by fusion of two unreduced gametes or duplication of somatic chromosomes followed by failure of mitotic division. Characters of autotetraploids. They show marked phenotypic variation, great adaptability, and show tendency towards perenniality. They are slower in growth and sometimes show disease resistance. They bear large, deeply colored flowers and fruits. They have large cells, mainly epidermal cells and guard cells. Allopolyploidy. Polyploid, which is formed by doubling of chromosomes of a F1 hybrid containing two distinctly different genomes, is called allopolyploid, and the phenomenon is called allopolyploidy, as you can see in the shown figure. Types of allopolyploidy. Allopolyploidy can be classified into three types, that is, genome allopolyploidy, segmental allopolyploidy, and auto-allopolyploidy. Genome polyploidy. Here, two or more distinct genomes are brought together through hybridization followed by doubling of the chromosome of the resulting hybrid. The F1 is often completely sterile. Chromosome doubling of the F1 hybrid produces an fertile allotetraploid called as amphidiploid. Here, example of Raphanobrassica has been shown to you. Segmental allopolyploidy. In allopolyploid, if some degree of homology exists between some chromosomes of one genome with chromosomes of other genome. Such allopolyploids are called as segmental allopolyploids. Autoallopolyploidy. Allopolyploids which show autopolyploidy for one or more of their genomes are referred as autoallopolyploids. Example, allodecaploid triticale. It has two copies each of the three genome of bread wheat ABD and four copies of rye genome R as shown in the figure. These are the references. Thank you.